Okay, what we're going to do today is talk about Unit 8, Genetics, and this is going to be the meiosis section of it, 8A. We're going to talk about genetic variation. This is the variation of genes, the parts of the DNA that you get from your mom or your dad, and the mixing of traits that everybody sees expressed in you. Here are a couple examples of animal and plant DNA variation, where we have 6, 7, 8, 9, 10, 11 giraffe, all of about the same size. Neck lengths will vary a little bit, but the most obvious thing is the coloration and the pattern differences. And then we have a picture of carrots, where we actually get variation in plant species as well. And meiosis, or meiosis, whichever you prefer, is when we actually get double cell division, where we take the sister chromatids from each of the homologous pairs, one from mom, one from dad, and we actually separate those so we can produce them into egg, the female gamete, and sperm, the male gamete. Here's a little diagram of it, where we start with the parent cell. If you take a look at it, we have basically two homologous pairs. We have the red ones and the blue ones, and when we actually mix them up, we go through prophase, and you can see them in there. Metaphase 2, they line up, and anaphase, where they actually change, but the difference is we actually have the X chromosomes on both sides, and we'll talk about why this is blue and why that's red, which we didn't talk about when we did regular mitotic division. And we'll go through another section where we go prophase 2, we had 1 and 2, metaphase 2, where we actually end up with a haploid section, where if this was a female, we'd end up with one egg with all the cytoplasm in one, and what are called 3 polar cells, which are sterile. If this is male, we get 4 sperm cells, or 4 male gametes. Gametes are the sex cells. They're either eggs if it's female or sperm if it's male, and you can take a look at it and you see the male. If this is a human, we got 23 chromosomes here. The 23rd chromosome, the sex chromosome, determines whether it's a boy or a girl. And this is an egg with also 23 chromosomes, the 23rd chromosome being an X. And if we get an X and an X, we get a female. If we get a Y and an X, we get a male. Fertilization happens when the male gamete, the sperm cell, reaches the egg cell, and that's the first one in, or at least the first one that has the enzymatic reaction to break through the egg and to spill the 23 chromosomes into the other 23, giving 46 for the human. And then we have a zygote, which is the fertilized egg. Here's after the first mitotic division, maybe after 4 or 5 mitotic divisions. And then we have cells in the testes, the sperm, and the ovaries, the egg, and we actually change it into a fertile egg and a fertile sperm, and then we take those into fertilization, and then we end up with the zygote, or the baby in case of a human, if we get all 46 of the chromosomes. And then here's a picture of an egg with a whole bunch of the male gametes around, all of them trying to vie for the fertilization. It is the enzymatic reaction of the winner who actually spills that one set of 23 chromosomes into the egg to get the fertilization process. And then again we go through mitotic divisions at that point, where we actually start getting different skin cells. Eventually we may get differentiation, where this one may become the heart and this one may become the brain, and different ones becoming different organs. And then we end up with a fetus, a zygote or a fetus in this case. And then here's a YouTube video which you can watch and actually see as this developing fetus, who had a tail at one point and gill slits at one point, actually gives us some idea about evolution, how maybe we got to the point where we are today. When we talk about meiosis, we have to talk about diploid and haploid cells. Diploid cells are also known as 2N. These are normal cells. It's when a human has 46 chromosomes, so you get the 23 from the male and the 23 from the female, and they are homologous pairs. We There's another section here about the strawberry. We just finished the strawberry DNA lab where we actually saw octoploid, where we're 2N, they would be 8N. Way too many chromosomes. Then we have homologous pairs, which are chromosomes that have the same information, like these two right here. One from mom's father and one from mom's mother. And again, father and mother, and then dad's mother and father, mother and father. And then when they break up through meiosis, we actually end up with a haploid set of cells, where we get this one from mother's mother, the grandmother, and the grandfather, and then maybe grandfather and grandmother from the dad's side, and then we get fertilization, where you can see mom's chromosomes and dad's chromosomes, who they inherited from their parents. Those are called homologous pairs, these two pairs right there. These pairs, these singles making a pair of homologous chromosomes. Here's another set of sister chromatids making a homologous chromosome. But basically they're chromosomes with the same information. And haploids are half this, where we actually get half the cells of a normal creature, which is also called N. In this case, it would be 23 for humans. And this describes sex cells, eggs and sperm. Haploid cells made through meiosis. So here you see meiosis, and we have two sets of it. These are diploid cells. We actually break those into single sets, which we call the haploid cells, and then we get fertilization, where we go back to diploidism. Tetrads are when we start talking about homologous pairs. So this one right here, where we actually talk about the fathers and the mothers, this would be called a tetrad. Tetrad are 4 chromatids, or the homologous pair of chromosomes. Here is a tetrad, and it actually has a chiasma, and a chiasma is a point where the 2 chromosomes may actually touch and swap genetic information. This is what's called crossing over. It's when two tetrads change genetic information. So if this was your dad's and this is your mom's, your dad's chromosome actually switches and gets the bottom of your mom's, and then your mom's will get the bottom of your dad's, and it actually increases genetic variation. This is how we can actually have individuals. Here's actually a very complicated section where we get double chiasmas, and we actually can see that you end up with a red and a green, that red and that green. We end up with an orange, a yellow, and an orange, a yellow, an orange, and a yellow, and a green and a red. Very much genetic variation going on there. Here's an example where we have paternal, where we can actually end up with a mom and a dad, where we can end up with quadruplets. If this one was an egg, another egg, another egg, and another egg, and we'll end up with those. Let me go back here, because I hate when I click that. You can see where we actually have an egg, an egg, an egg, an egg, and we get 4 different sperm for fertilization, sperm cells for fertilization, and we can end up with different genders and also different looks in the genders. Here is the basic idea of meiosis. We have meiosis 1, same phases, interphase, prophase, metaphase, and anaphase, where we actually take it. The difference is when we get chiasmata, we can get some genetic variation, and when we start splitting it, instead of actually getting sister chromatids, we actually split the individual chromosomes, and you can see we've shifted some of the information from the red to the blue and the blue to the red. So we actually get chromosomes duplicate, we get homologous chromosomes pair up, and we get tetrads lining up, 2 sets of X chromosomes, and we get the pairs of homologous pairs actually splitting up. Then we move to meiosis 2, where we actually get the cleavage furrow, and we get the individual chromosomes moving over, and then we're going to get those prophase 2, where we actually start forming them with the centrioles and the spindle fibers, and then we get metaphase 2, where we actually get them to line up, and it grabs on to the centromere, and it actually now is going to split them up, sort of like and actually meiosis 2 is very much like mitosis, but now we're splitting them up, and we're going to end up making what are called haploid cells, where we actually have half the information in each one of the cells. And you can see that now that we have 4 of them, we have 4 haploid cells, which can actually go through and fertilization and actually make a diploid organism. Okay, we're going to do an assignment, cutout and matching activity, but this is actually going to be SNO assignment 4, where the kids are going to go through and actually learn about 8A and hopefully take notes on it in their PowerPoints, which they don't have, but I'm hoping they'll take notes on notebook paper. Appreciate you stopping by. Bye.